podcast number 34, where we're going to look at another emergent property of cells, and that is memory formation. So we have actually a pretty good idea about how memories are formed at a cellular level after years of research, and I'm going to go over some of those experiments in this podcast. A lot of this work was done with this organism, which is a sea hare, also called a sea slug, also called a nudibranch, and the genus is Aplysia. And I'm going to show you a video of one of the scientists who pioneered this work, and some video of Aplysia here.
The Aplysia stretched out. We're going to focus in on the gill and the siphon. We're going to apply an extremely weak.
So Aplysia is a ocean animal, marine creature, lives in the ocean. It's very much like a snail without a shell. It breathes through a gill. This is its gill here, and it takes in water to bathe that gill so it can breathe, and then spits that water out. Now I'm going to show you some pictures where they're giving this animal a stimulus it doesn't like. So they're going to poke at this mantle, which is sensitive, and it causes them to retract their gills as if they are going to, as if a predator, like a fish, might be going after that. And then they give it a slightly stronger stimulus, and then you're going to see these electrodes giving it a slight shock. It's still a very weak stimulus. I'm sorry if this bothers anybody. The animal is not greatly hurt by this. This is sort of like stubbing your toe.
Stimulus, so you can see the amplification with sensitization. Weak stimulus to the siphon, you see a modest withdrawal of the gill. Now we're going to frighten the animal, startle it, give a noxious stimulus to the tail. That, of course, causes a contraction in its own right. This contraction lasts for seconds, but you can come back minutes later. The same siphon stimulus now produces a much more powerful withdrawal. We can now compare the two, normal and sensitized, and see how much more powerful the withdrawal is in the animal that is startled. The memory for this event is a function of number of training trials. So if you give a single tail shock, plotting here, change in reflex strength is a function of time, the enhancement of the reflex lasts minutes. But if we give the same stimulus 4 or 5 times, we get a memory that lasts a couple of days. So we have a short-term memory and a long-term memory, and we can now look at the difference between them.
Okay. So it's pretty surprising that what we consider a simple organism here forms memories. Maybe not so surprising, but we tend to think of that as a higher order function, certainly an emergent property. One of the big ideas of this book of you put a bunch of cells together and you can get memories in there. And this sea slug is definitely remembering being stimulated. Again, from that video, if there has been no learning of sensitization, nothing much happens. If they give one lesson, they get some response. Number of lessons over a few minutes, you get a short-term response, and if you spread that over days, you get a bigger response and a long-term memory. So what's the difference between short-term and long-term memory in terms of at a cellular level? And this is the system that they use to figure that out. So in this organism, it has ganglia, not so much a brain, so to speak, but just collections of neurons, and not a whole lot of neurons, but enough so that they could label them and repeatedly measure them. So they labeled them according to the left side of the body or the right side of the body and gave one each number, and then tried to map out what they were doing. And he and his workers characterized different types of neurons. So there were sensory neurons, neurons that were detecting a stimulus, like getting poked with a pipette. Motor neurons that were going to transmit a stimulus to the gill and cause it to retract. So like you would stimulate a muscle and cause that to retract. And they worked on how these neurons are communicating with each other by recording from different cells at the same time, try to figure out what was going on. So they were able to draw these circuits after doing lots of experiments. And they were able to quantify how much the gill retracted. So here's an outline of some of the experiments they did. So they took the L7 neuron, so this neuron number 7 on the left side, which is a motor neuron, and they stimulate that. And then they can measure that after they stimulate that, the gill will retract. So there's a connection between that motor neuron and a gill reflex. And then they can stimulate a sensory neuron, get a reaction in a motor neuron, and if they give several spikes, they get a stronger reaction, so on. So in this simple learning circuit, simple compared to what is happening in your brain, they could measure that once they give tail a shock, there's a connection taking that information through an action potential to another, making a synapse with another neuron that's going to release a neurotransmitter called serotonin. You have serotonin also. That's neurotransmitter in this neuron then makes a connection with a sensory neuron, which makes a connection with a motor neuron, which causes the gill to retract. So depending on what part of the circuit you stimulate, you can measure different sorts of behaviors and then measure whether or not learning has happened and how that learning has happened. So they can give a training stimulus applied to the tail. They can say that learning takes place in the sensory neuron. And then they could also distinguish between short and long-term memories. So they were able to establish that short-term memories happened first before long-term memories. And in short-term memories, what happens is after a stimulus, there's a release of serotonin in this particular pathway. It doesn't mean that serotonin is the neurotransmitter in all learning pathways, but in this particular pathway, okay, that sensory neuron gets excited and releases serotonin. Serotonin binds to a receptor that interacts with the G protein, just like when we talked about epinephrine interacting with this G protein. That G protein interacts with adenyl cyclase, just like you saw with the fear pathway, and cyclic AMP is produced, just like you saw in the fear pathway. Again, just like you saw in the fear pathway, protein kinase A is activated by cyclic AMP. Protein kinase A binds to cyclic AMP, and that turns protein kinase A on. Protein kinase A is a kinase, so it's going to take ATP and add phosphates onto other substrates. And then that synapse is modified by calcium and the protein kinase A. So calcium will enter the cell from outside, higher outside than inside. It interacts with protein kinase A, and it modifies the synapse in that it is now more responsive than it was before this happened. So if we go back in time here, so that protein kinase A is now going to modify this synapse, and so it's going to be more responsive to interacting with the motor neuron down here. Okay, and these are synaptic vesicles piling up on the face that is facing the motor neuron. Okay, that's short-term memory. Okay. And this is after a single exposure, and this would not last very long. So this protein kinase A would get turned off by a phosphatase, the cyclic AMP would degrade, the G protein would switch out GTP for GDP, adenyl cyclase would stop making cyclic AMP. So this is a short-term effect, lasting minutes to hours. If, however, the stimulus is prolonged, so this is the graph where they gave the stimulus repeatedly over 4 days, now protein kinase A starts to stay around long enough that it starts to phosphorylate other sorts of substrates like MAP kinase. MAP kinase was part of that kinase cascade that you saw in the fear response. That one thing phosphorylates another thing, and protein kinase A, if it stays active long enough, like with repeated stimulation, can activate MAP kinase, which is another kind of kinase. MAP kinase will then travel to the nucleus, and in the nucleus it will modify a protein called CREB, which stands for cyclic AMP responsive element binding protein, or CREB for short. So cyclic AMP responsive element, it responds to cyclic AMP, and it's a binding protein. It's actually going to bind to DNA. And when it binds to DNA, it activates the transcription of lots of genes. Okay? And then these include some exocytosis proteins, proteins that are involved in causing more neurotransmitter to be released. And the synapses get enhanced. So if you can see before long-term potentiation or long-term memory. Let's go back in time here. Okay, you have synaptic vesicles missing here. Okay, and then after long-term memory happens, you get more synaptic vesicles showing up there. So in this system, which is a really useful system because it was pretty simple, they were able to distinguish between what sort of cell changes happen that cause short-term memory and how are those cell changes different from long-term memory. So some of the experiments they did, for example, was let's do some long-term memory sort of training where we stimulate the animal over several days, but then let's stop the animal from being able to make new proteins. And when they stop the animal from being able to make new proteins, they don't get any long-term memory. Because if you stop it from making new proteins, CREB might be able to transcribe these genes, but then they never get translated into proteins that can actually change the synapse. So you got to have protein synthesis. You don't need protein synthesis for short-term memory, but you do for long-term memory. Some takeaways that you can take for this in that the reason that it takes repetition for you to remember something is that that repetition is required to activate protein kinase A for long enough inside your cells to then activate MAP kinase, to then activate CREB which then will turn on genes that were not turned on during short-term memory. And more training strengthens the synapse even more. The synapses and the neurons physically change as a result of this long-term memory training. So low density of synapses, each synapse here is this little bright spot along this axon here. And then, higher density of synapses after long-term memory, and then at very high density of synapses after reinforcement of long-term memory. So the neurons physically change as a result of the proteins that get turned on as a result of continually getting stimulated and continually reinforcing that synapse. So this doesn't necessarily tell you why, as all aspects of memory is a complicated thing, but this is a relatively simple system where some pretty complex rules about memory were established, like the difference between long-term and short-term memory, and what is required, and that protein synthesis is required for long-term memory. These sorts of experiments have been repeated in mice, and these results have held up. And the scientists who developed this system in Aplysia and the sea slug won a Nobel Prize for this work. So it's another example of how you can go from basic cell biology and how cell signaling works to an emergent property, something like a memory, and also why repeating something when you're trying to memorize something works.